Alright. It should be live now. So, might want to go ahead and adjust your volumes so that I don't sound like I'm blaring through your headphones or speakers or what have you. We're going to be holding this until 9 o'clock tonight. Please feel free to submit your questions either through the social stream there, as you see Yulin Chen has demonstrated so aptly, or just easier to send them to me through email. Got a couple questions to start us off with, but yep, just go ahead and keep sending them to me, and I will keep checking my email. So let's get started. Start with Yulin's question. Okay. So, I'm confused about the functions between voltage-gated sodium channels and voltage-gated calcium channels, and what's the different functions between them. So remember, voltage-gated sodium channels are present on the axon hillock, and those voltage-gated sodium channels open when the inside of the neuron reaches a certain threshold, and then that lets in a huge amount of sodium, and that sodium influx is what starts that depolarization that begins the action potential. Voltage-gated calcium channels are very similar in that they also open when the neuron reaches a certain threshold, but the only difference is they let in calcium. So, insofar as the different function, like which ion they let in, they're the same. They're both channels that are just opened by the membrane reaching a certain threshold. Sodium channels let in sodium, calcium channels let in calcium, and the sodium channels are responsible for the initiation of the action potential, whereas the calcium channels, that calcium influx, is one of the signals on the presynaptic membrane for there to be neurotransmitter release. So remember, calcium is very low in the presynaptic membrane, so calcium goes in, that's a signal to release neurotransmitters. So that's the difference between those two. If you have a follow-up question, just go ahead and shoot that over. Alright, got quite a few more. Let's take a look here. Alright. Yes, it is much easier if you email them to me, just because I have them here on my phone so I can look through them. What happens to the pattern of action potentials when higher levels of electrical current are introduced? Also, what are the functions of the mitochondria that you would like us to know? Okay, so what happens to action potentials? So say you have a current. Well, what happens to, I think that's pretty much straight off the study guide, so this will be good. So say you have a current here, and you have a current that's, level this, and that'll just initiate an action potential, like so. What happens when you increase the current, however, is you increase the frequency of the action potentials. So they're going to fire more frequent. No, they're not going to be bigger. They're not going to have a higher amplitude. They're just going to fire more rapidly. So you could imagine if you increased it even further, then you get even quicker firing. So the intensity of the stimulus here is encoded in the frequency of the action potentials. Frequency in electrical terms describes the number of times that a signal occurs within a specified period. So, increased current causes an increase in the frequency of the action potentials. Alright, so what are the functions of the mitochondria that you would like us to know? I'm really not too concerned with the mitochondria. Mitochondria do cellular respiration, of course, so they provide ATP for the cell. So they're not directly related to anything. They just sort of provide this energy source for everything inside the cell. That being said, everything that happens because of the presence of ATP, but they do not have a direct impact on like building proteins or transportation of neurotransmitters or what have you. So if you're familiar with them insofar as they just have this broad function of providing energy for the cell, that will be sufficient. Alright. Moving right along. Lots of questions, good stuff. When you say identify the spike initiation zone, do you mean be able to label it or know the definition? What I mean by that is be able to label where it would be on a cell. So you know that one of the presentations you could see that it's at the axon hillock. It's where all the potentials summate and the action potential either fires or it doesn't. In the sensory neuron, it's just a little bit sort of to the left. It's down on the left portion of the axon. Don't worry about the distinction between those two. Basically, knowing that the spike initiation zone is where the axon action potential starts, and that it's at the axon hillock will be more than sufficient for the purpose of this exam. Okay. Question on G protein coupled receptors. Still confused about G protein coupled receptors, how they work, what they do, what makes them so special. Could you please explain them again? Yes. Yes, I can. Alright. So, incidentally, I am recording everything that's going on here. So, if anybody's feverishly taking notes, I'm not going to stop you. But if all things go according to plan, I should be able to post this online on YouTube or something and then just send a link. However, we know how the first recording attempt went with the audio, so it might not be a bad idea to take notes. Okay. G protein coupled receptors. So remember, there are two different kinds of receptors that we talk about. So I'm just going to illustrate. Here's a membrane, phospholipid bilayer, and in that membrane is an ion channel here. So what happens when an ion channel opens? Well, what can make an ion channel open? A lot of things. You can either have, the inside of the cell reaches a certain voltage that causes a conformational change, and then the ion channel opens, allows ions to flow in and out. Sorry for all the coughing. Or you could have something that comes in and binds to the receptor. And it comes in and binds here. And then that causes the ion channel to open, and you can have ions that flow in and out. So what happens with an ion channel is when that opens, say that this is a sodium channel. When it opens up, you can get sodium that's going to flow into the cell. If it's a sodium and potassium channel, you can get sodium that flows into the cell, and you can also get potassium that flows out of the cell. Or possibly even potassium that flows into the cell. So that's what ion channels do. They open up and they allow for the flow of ions in and out of the cell. And remember, when ions flow in and out, that causes increases or decreases in the voltage of the cell, and that results in either excitatory postsynaptic potentials or inhibitory postsynaptic potentials. Right. So, that's the function of just an ion channel here. It's very boring, very simple. So then we get to something like a G protein coupled receptor. So here we'll draw that in the membrane. And I'll just draw it like so, similar to what was in the book. So what happens here is you have a receptor similar to how you had in the ion channel here. So you have something that comes in and it binds here. Let's say it's a neurotransmitter. But then what happens is this channel does not simply open. What it in effect does, what the G protein coupled receptor does, is it initiates a whole sequence of downstream effects. So what there really is there's this complex here. We're not going to get too bogged down in the specifics. But when this is activated, one part of this complex shoots off this way, and the other parts, well, the other part will go the other way. And these will, when this complex, this is like a messenger. It'll just split off here, and then this part will split off here. And it goes down and it'll activate other enzymes in the membrane. So when the G protein coupled receptor is activated, it sends out these messengers, and then they'll activate enzymes. And those enzymes will do something fancy. And then that will go and activate another enzyme. Okay? Then that'll do something fancy. Then that'll activate another enzyme. Keeping this pretty simple here. I'm not worried too much about the specifics. But basically what the G protein coupled receptors do is they will start this chain reaction in the cell, where instead of just doing something relatively boring like letting ions flow in and out, they'll start activating all of this intercellular machinery. And the end result of that is that basically a ton of stuff can happen. These are called second messengers. This is what we're going to call second messengers, is these different steps in the G protein coupled receptor pathway. So what these second messengers will do is they will do something called modulation, and they can also do things like phosphorylation. Okay, so what is cool about these? Well, again, what are you going to get when you open up an ion channel? You get an increase in voltage, you get a decrease in voltage, or you make it more difficult to initiate an action potential. That's interesting, but when you activate a G protein coupled receptor, you can have phosphate groups that will bind to ion channels, and that will affect the action of the channel itself. It can affect, how many ions flow through. It can impact the efficacy of the channel. You can also have little chemicals that will attach to other ion channels here that are downstream products of just some enzyme chain. And that'll put more, and that'll indicate to the ion channels that they should go up to the membrane. So you can actually, by the activation of the G-protein coupled receptor, not only can you get a modulation of the activity of these ion channels, but you can get more ion channels that go into the membrane itself. And then they can do a whole ton of other stuff too. So, to answer your question, the reason that they're cool is they don't just let in ions. They don't just change the voltage of the neuron slightly. They let in a whole, they start a huge chain reaction that has these massive effects on what's going on inside the cell. It can influence what chemicals the cell is making. It can influence the number of ion channels in the membrane, and it can influence the efficacy of channels that are already in the membrane. So the effects of G-protein coupled receptors are much more substantial than just simple ion channels in that respect. You do not have to memorize any of the second messenger cascades that go on inside the cell, but just to get sort of back to the mitochondrion question, a lot of these reactions with these enzymes require ATP or protein kinase or just different things like that. So it actually is kind of complicated what happens here to here to here, but all you need to know is G-protein coupled receptor gets activated and a whole bunch of other stuff happens. It will change a lot of what the cell is doing and what the ion channels in the membrane are doing, and that it will be much more substantial than just activation of any one ion channel. If I haven't answered any of your questions, just email them to me again, and I'll give them another go. Alright. Next question. I'm confused about this process. Describe the process of how an action potential causes a release of neurotransmitters. Be sure to use the key terms presynaptic membrane, voltage-gated calcium channel, synaptic vesicle, exocytosis, neurotransmitter molecule, synaptic cleft, postsynaptic membrane. First, can you describe this? And second, does this happen every time there is an action potential, or only in certain cases for certain ions? Alright, give me a second. I just want to read this over again. Alright. This is basically one of the questions in the study guide, but we can answer it nonetheless. Okay, describe the process of how an action potential causes a release of neurotransmitters. So, we'll start with the key terms. So, presynaptic membrane. Remember, in a synapse you have both a presynaptic membrane, illustrated here, and you have a postsynaptic membrane, which I've illustrated there. Synaptic membrane. Okay, and in the presynaptic membrane, you have these little ion channels that are voltage-gated calcium channels. Alright, and you also have, remember in the presynaptic membrane you have these vesicles that are filled with neurotransmitters. So, synaptic vesicle. You get the idea. So, what happens with an action potential, and how does that cause a release of neurotransmitters? Well, remember we talked about the action potential propagating down the axon, and eventually it'll arrive, the action potential, let's say that the axon is leading up to this particular presynaptic membrane. So this is an axon terminal, in addition to being a presynaptic membrane. So you have an action potential, it's going to go, travels all the way up here, and then eventually it will reach these voltage-gated calcium channels. And when the action potential is traveling up here, it'll cause the inside of the cell to become more positive. So let's just say that it's depolarizing the cell to positive 20 millivolts, just for the sake of argument. So, usually the cell is at negative 65 millivolts, but wherever this action potential is propagating, it goes up to positive 20 millivolts. So, when the action potential reaches the voltage-gated calcium channels, they'll respond to the voltage, the inside of the cell being so positive, by opening up. And what happens when the voltage-gated calcium channels open, just for the sake of being perfectionist, calcium is going to flow into the presynaptic membrane. So the action potential propagates down here. Then it will open up these channels, and it will allow calcium to flow into the presynaptic membrane. When that happens, remember that the inside of the presynaptic membrane has a very low concentration of calcium. So when that happens, calcium is detected pretty quickly when it goes into the cell, and it is a signal for the synaptic vesicles to go down into the membrane here, and then they fuse with the membrane. And when they fuse with the membrane, they open up and they spill out all of the neurotransmitters that are inside them. And this process is called exocytosis. Now remember, in one of the talks today, the person was talking about spider venom. What that actually does, incidentally, is as she described, it stops the process of exocytosis because it actually interferes with the proteins that fuse the synaptic vesicle with the presynaptic membrane. But you don't have to worry about that. It's just an interesting point. So, action potential goes down here, hits the voltage-gated calcium channels. They open up, calcium goes in. That signals a synaptic vesicle to go into the membrane, and it releases its contents, the neurotransmitters, in a process that's called exocytosis. I'm going to label these neurotransmitters. Okay, so those are the neurotransmitter molecules, and then what those do is they will diffuse across the synaptic cleft here, which this is getting a little bit crowded. But this space in between the presynaptic membrane and the postsynaptic membrane is the synaptic cleft. And these will diffuse across the cleft, and they will activate receptors on the postsynaptic membrane and cause those to open, and then ions will go through. Sodium in, potassium out. So that is the process of how an action potential causes a release of neurotransmitters. In short, it depolarizes the presynaptic membrane, opens up voltage-gated calcium channels, and then that begins the process of exocytosis, because the presynaptic membrane will detect calcium, which is in a very low concentration normally, so that's the indicator that it should fuse the synaptic vesicle with the membrane and spill the neurotransmitters all over the synaptic cleft, or diffuse, I should say. Okay, so first, can you describe this? I just did so. Second, does this happen every time there's an action potential, or only in certain cases for certain ions? I think that should say certain cases for certain neurotransmitters, but I'll answer it either way, just to be sure. So does this happen every time there's an action potential? In theory, the action potential should propagate down to the axon terminals and cause a release of neurotransmitters. So, yes, in theory it should happen every time there's an action potential. In practice, every action potential does not reach an axon terminal because of various things that we're not going to worry about right now, but suffice it to say that not every action potential ends up becoming neurotransmitters being released. And does that happen in only certain cases for certain ions? So, to answer the question for only certain cases for certain neurotransmitters, I mean, it depends on the presynaptic membrane and which neurotransmitters it has in it. So if it's a cholinergic cell, then it'll release acetylcholine, or noradrenergic, noradrenaline, all that stuff. So it's pretty ubiquitous in terms of the neurotransmitters it releases, at least insofar as we're concerned with it. For certain ions, let me think of how to answer this. So, again, I just don't want to make this complicated. It's not selective for any particular ion. An action potential propagating down the cell, in this case, specifically activates or allows the influx of calcium, but I think that you don't have to worry about ion specificity in this case. I think that answers your question. If not, just send me another email and I'll give it another shot. I just got an email. Meant neurons. Neurotransmitters. Okay, great. So that's the answer. All right. Okay, what is the function of the Golgi apparatus? Oh my, this is a long email. Can you describe the amount of detail that we need to know in these questions? Describe the process of how an action potential causes... Alright, these are different questions. What's the function of the Golgi apparatus? So, the Golgi apparatus is... Basically, it is responsible for post-translational processing of proteins. Now, what does that mean? Proteins are translated or constructed from RNA in the endoplasmic reticulum. The analogy I used is that in the endoplasmic reticulum, it's like the little elves are building the presents for Christmas. They're building all the toys. In the endoplasmic reticulum, they're actually building the proteins from the blueprints that they have um, that are provided by the RNA. What happens in the Golgi apparatus, or the Golgi body, is where they wrap the presents up, where they put bows on them, and where they put mailing addresses on them, and then they get shipped out. So, you do not need to know the specifics about post-translational processing, but that's the distinction. What happens is just dotting all the I's, crossing all the T's, putting a bow on it, doing the final changes and the final additions to the proteins before they're shipped out to wherever they're going. That's what happens in the Golgi bodies. For the exam, if you're familiar with what's going on without it being any more specific than that, you should do perfectly fine for our purposes. But yeah, that's what the Golgi body does. Endoplasmic reticulum builds the protein. Golgi body does the post-building additions. Alright, can you describe the amount of detail that we need to know these questions? Describe the process of how an action potential causes a release of neurotransmitters. I already answered that, but I'll talk about the detail in a second. Function of a G-protein coupled receptor and how this differs from an ion channel. Modulation. Okay, so the amount of detail that you need to know these questions. If you can write a sentence or two that uses all of the key terms that I put in there in bold, so for the release of neurotransmitters, that's presynaptic membrane, vesicle, exocytosis, synaptic cleft, postsynaptic membrane. If you can describe the process using those key terms, you should be fine in terms of how much detail you need to know. It does not need to get much more complicated than that. Remember, this is multiple choice, so what I'm likely going to ask you is either what's going on at this particular, I'll give you a figure and say, what's going on at this particular phase. Or I will, give you a key term and say, how is this involved in this? And you'll get to choose among all the answers. So, knowing the key terms and knowing how those fit in should be a sufficient outline for the detail that's required. G-protein coupled receptor. So I also answered this question earlier. So again, what's a G-protein coupled receptor? It is a receptor that is tied to a bunch of enzymes on the inside of the cell that cause these diverse effects, not just letting ions in and out. And those diverse effects are the product of second messengers and second messenger cascades, and that's just one enzyme going to the next, going to the next. And they can do things like modulate the activity of either different proteins on the inside of the cell. They can modulate the activity by activating enzymes, or they can modulate the strength and efficacy of channels in the membrane itself. And so, you know how that differs from an ion channel because I described that. An ion channel is just a channel that opens up and allows ions to flow in and out. So again, if you can describe the function just using the bold key terms, that should be sufficient. Then correct steps for the dorsal column medial lemniscal pathway. Somatic sensation, let me just read over these real quick. Alright, yeah, so here, I'm going to draw out, the person who sent me this email, you have it correct, basically, with the dorsal column medial lemniscal pathway. You don't really have to know it in any more detail than what you described here, but I will draw that out for everybody so that they can see. Really, actually, I mean, the detail with which you need to know it is what's present on that one PowerPoint slide, but I'll just draw it right here. So you have, say, you have some skin here. Then you have something that comes in here and pokes the skin. So the movement in the skin is detected by mechanoreceptors underneath the skin. So here we have mechanoreceptor. And what that does is it's mechanoreceptor communicates the information to sensory neurons in the peripheral nervous system. So it's going to talk to, well, it's also, I believe it's a kind of sensory neuron, but we'll just call it sensory neuron. And really, if you conflate these two, it's fine. The first step is sensory neuron detects a sensation on the skin. I'll draw this as a hand, so you know what that is. So sensory neuron detects a sensation. So then what that's going to do is it is going to communicate that sensation all the way up to the spinal cord. As we know, sensory information travels up the spinal cord in the dorsal pathway. So here we have our spinal cord. Here you have the horns. I'm not going to worry about that. So the information goes from the periphery to a sensory neuron and then onto the spinal cord right here. So first step is sensation, feeling it with a sensory neuron. That sensory neuron sends the signal up to the dorsal column of the spinal cord. So then what happens? It'll travel up the spinal cord, so we have our spinal cord here. That information will travel up the spinal cord, up, until it gets to the medulla. Now, it's going to have this traveling up, like so, and then we're going to zoom out a little bit. So here we have the brain, the cerebellum, and the medulla, and the brainstem. And part of the brainstem here is the medulla. And then you also have, this is connected to, well, more or less the spinal cord. So here's our central nervous system. Yeah. So what happens is it travels up the dorsal column pathway, and then it will get to what is called, it's going up the dorsal column pathway, and then we're focused right here on A, and it'll get to the medulla here, and it'll go into the dorsal column nuclei. Dorsal column nuclei sounds more complicated than it is. Nuclei, or their nucleus, just describes a group of similar neurons. So what happens here is the dorsal column nuclei might as well be called, like, gate 4A for United Airlines or whatever. It's just that's the receiving area for information coming up from the dorsal column of the spinal cord. So then it's received at the medulla in the receiving area, and it will travel across the medial lemniscus. in the medulla. So this is just another part of the medulla where it goes across. So it's shifting from one terminal to another, and then it's going to get on its connecting flight, and its next step is going up into this part of the brain, which is the thalamus. So here, I'm actually going to pull back here. So here we have B. What happens here, we'll go medial lemniscal pathway, is that this will then travel from the medulla into a specific nucleus of the thalamus. Now remember, the thalamus is this sort of central structure in the brain here, where a lot of sensory and motor information is communicated to and from the rest of the cortex. So you have the very complicated cortex with all of its intricacies and psychological functions, and the way this speaks to the brainstem, and the way the cortex speaks to other regions as well, is by going through these sort of these nuclei within the thalamus. Again, remember, these nuclei are just bundles of cells that have similar function. So, there's a nucleus for auditory information, and there's a nucleus for visual information. And this happens to be a nucleus in the thalamus for sensory information that's coming from the medial lemniscal pathway. So that travels up into the thalamus, and it hits that special nucleus for the sensory information. The thalamus will then send all of that information, because its job is to send it into the cortex. It will send it into corresponding regions of the somatosensory cortex. So this is the brain. This is the wrinkly brain. This is part C. And these different somatosensory regions will represent different parts of the body. And in the case of our hand getting pricked here, the thalamus is going to interpret the information coming up from the spinal cord that went through the medulla, over the medial lemniscal pathway and into the nucleus. It interprets that information and says, I know, this is you getting poked in your hand. So then it's going to send out that information to the parts of the cortex that represent the hand, the parts that represent the arm, the parts that maybe represent, the wrist. So the different steps here really are, there's a sensation step where the information goes to the spinal cord, and then there's just A, B, C. A, medulla, B, thalamus, C, cortex. So sensory information is perceived by sensory neurons. It travels all the way to the spinal cord, it goes up the dorsal column, and after it's traveled all the way up, it'll hit the medulla, at which point it crosses the medial lemniscal pathway. Then it will go all the way up to the thalamus, and then the thalamus will send that information to the rest of the brain. So let me just draw that again. That was quite a messy drawing. So just here we have spinal cord, brainstem, and brain. And the spinal cord has sensory neurons that go into the hand with too many fingers and a tumor there apparently. So proprioceptive or sensory information comes in here, travels up the sensory neuron into the dorsal column of the spinal cord. That information travels up, till it hits the medulla. It goes into its little terminal, the dorsal column nucleus, goes to the medial lemniscal pathway. Then it will continue up, 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 until it hits the thalamus. Then once it goes to the thalamus takes it and says, alright, this is the hand information. I'm sending that to the portions of the primary somatosensory cortex that correspond to the hand. That little rockily piece of hand there. So that is the detail with which you need to know the pathway. Sensory neuron, medulla, medial lemniscal pathway, thalamus, primary somatosensory cortex. And again, remember, you're not being asked to list these by memory. You're just going to have to be able to identify these different pathways as they occur naturally in an image. That is a terrible image, so I'm going to go ahead and delete that. But we will move right along. I might need to take a 5-minute break or so at 8 o'clock, just because my throat is getting dry. But we will cross that bridge when we come to it. Does not look like we actually have too many more questions. I have 2 more here. So if you have any more questions you'd like answered, please send them in. If I don't get any questions for half an hour, then we are done. Okay. What are the different types of glial cells, and what is their function? Okay. I'm actually, because this is just directly related to something on one of the PowerPoints, so I'm just going to pull that out and I'm going to show it to you, because this is all you need to know about. All right. All right, here it is. This is all you need to know about glial cells. There are basically 3 different kinds of glial cells that we're concerned with. There's astrocytes, there's microglia, and then there's myelinating glia. Astrocytes and microglia have sort of a similar function. Astrocytes regulate extracellular space, so this has not only to do with like trash and debris and whatnot, but also the ionic, the ions that are in the extracellular space. Hold on one sec. So I'm getting word that nothing can be seen right now. So I'm going to try and fix that. And stand by. Okay. Alright, I'm going to stand by. We're just running into a couple technical difficulties. Alright, I'm going to restart the stream really quick, so it's going to go down for a second, and then in about 2 minutes or so, just try refreshing, and I'm just going to use this as an opportunity for about a 5-minute break. So I'm going to get back at 8 o'clock p.m., so if you can hear me, the stream is down right now, but you should be able to hear my voice, and I'll just be jumping back at 8 o'clock, so come on back then, and I'll see if I can't get this resolved by then.